Application number 1. Karen's technique for measuring the length of DNA molecules by audio radiography. John Cairns is credited with measuring the length of the E. coli genome using autoradiography. This technique was normally used to test for the presence of substance in tissues. However, Cairns used the same technique for a whole new purpose. Cairns grew bacteria in titrated thymidine, which contained the radioisotope tritium. After 2 generations, it was fully integrated into E. coli's DNA. Radioactive substances, like titrated thymidine, break down over time and release particles. The cells were placed on a dialysis membrane and treated with lysosomes that digested the cell wall. Then, the cells were burst, which released their DNA onto the membrane surface. The cells were covered with photographic emulsion, which could detect electrons emitted by the radioactive DNA sample. After 2 months, he developed the film and observed circles representing the bacteria chromosome. Using the image, he was able to determine that the E. coli chromosome was 1,000 microns long. I guess that's not microns, that's nanometers? Nope, not nanometers. UM. I should know this. UM. It is a micrometer. Is a micron same as UM? I think it is. Micrometer. Also commonly known as a micron. Ha! I need to trust myself. Yes, 1,000 microns long. This technique was then used to visualize various eukaryotic genomes. Okay, so I'm going to go back over that whole thing and explain it in non-scientific ease, or at least try and make more sense of it. All right. So, Cairns grew bacteria in titrated thymidine. Remember, we have our base pairs A, G, T, C, right? A, G, T, C. So, what he's basically doing is he grew bacteria in a solution that has radioactive T, right? So, that means the DNA of the bacteria that he grew had, every time there was a D or a T base pair, right? Those were radioactive. It didn't necessarily harm the DNA, right? It just kind of like gives off radioactive particles. All right, so now you have bacteria with this radioactive DNA, right? So he took the bacteria and he put it on a plate, right? So this is a plate. And he added lysosomes, right? These are things that break down stuff. These are organelles that you should know about, right? Lysosomes. In animals, they break stuff down. And that breaks down the cell wall. So the bacteria basically spill their DNA out onto this plate. It's like spilling their guts out, sort of. Now, remember, their DNA is radioactive, meaning that energy is going to be emitted as those radioactive particles decay. So what he did is he poured a photographic emulsion, which is like basically the stuff on film that lets you take a picture, right? If you've ever, I don't know if you guys ever played with this when you were a kid. I used to get it for Christmas all the time. It's like photographic paper where you would like put stuff on it and then stick it out in the sun. And the parts where the sun shone would get really dark, and the parts that were covered up stayed light, and you could like make cool art with it, photographic paper. This is basically doing the same thing. So he pours this photographic emulsion over the spilled DNA guts of the bacteria. Now, those radioactive particles start emitting electrons and emitting radiation, which is recorded by the photographic emulsion. And what you end up with is a picture. So all the dark spots are where the radioactive T bases were emitting energy, and that was recorded by the photographic solution, right? So it looks like we have a big loop, right? And it's not solid because remember, those are only the T bases. So it looks like it got crossed over in a couple places. But that is basically the big chromosome in E. coli. And when he like mapped it out and then he could measure, it was about 1,000 microns long. Application number 2. Comparison of genome size in T2 phage, E. coli, Drosophila melanogaster, Homo sapiens, and Paris japonica, otherwise known as bacteria, E. coli, fruit flies, people, and Paris japonica. Grapes? Let's see. Nope, not grapes. A really pretty plant. The Andromeda plant. Okay. The genomes of many organisms have been studied in recent decades, and a wide range of sizes have been observed. Examples of genome sizes in base pairs. Now remember, a genome is all of the genes in a species. So all the alleles, all the types of genes. So the human genome would include all the genes for all different color types of eyes, right? All the possible genes in all the humans on the planet. That's the human genome. So T2 phage has about 170,000 base pairs. So like AT, that's a base pair. Or CG, that's a base pair. So T2 phage has about 170,000 of those. E. coli has 46 million. E. coli is a bacteria. Right? So is a T2 phage. Fruit fly. Fruit, I can spell. Fruit fly. 130 million. Homo sapiens, 3.2 billion. Plants, because they can do all those cool biochemical tricks that we can't, 150 billion. So, plant genome sizes tend to be very, very large because biochemically they're very complex and can do lots of things. Animals tend to be a little less complex depending on what kind of animal they are and how complex they are. Bacteria, a little simpler, right? And it makes sense. As the complexity of the organism increases, so does the genome. You can think of your genome as a deck of cards. Each card is a trick that you can do. The more tricks you can do, right, the bigger your genome. Here we have a chart that shows genome size differences both within and between different classes of organisms. So mycoplasma, I believe, is a parasite. Is it a blood parasite? Let's see. Mycoplasma. Isn't that the thing that causes... Nope, it's a bacteria. Right? Yep, bacteria that lack a cell wall, which means they're resistant to antibiotics that target cell walls. They can be parasitic or saprotrophic. I'm pretty sure they're like, yeah, Mycoplasma pneumoniae causes lung infections. Okay, yeah. So this is a bacteria that causes, this is a bacteria that causes lung infection, right? These are all bacteria. Now we have fungus, algae, and I just lost my screen. My computer does this thing where like The screen goes blank. It's still doing all the stuff it's supposed to. I just can't see it. So I'm going to pause right here and try and figure this out. Okay, I'm back. I fixed my screen. So we have bacteria here, molds and fungus, algae, and now we're getting into eukaryotes, right? And as we go down the list, we can see that there's a lot of variation in terms of the genome size of different organisms. We have worms that are pretty small. It's still, big, 10 to the 8th. But then you take a look at echinoderms, things like starfish, or insects, right? Lots of different kinds of insects. Their genome is huge. Mollusks like clams, birds tend to be pretty small. Their genome is kind of simple. Part of that is because these guys evolved from dinosaurs, so most of their genetic diversity was lost at the bottlenecking event after the loss of the dinosaurs. Or I guess that is the bottleneck effect, right? When you lose a vast amount of genetic information. Bony fish, they've been around for a long time. So they have a pretty large genome. Cartilaginous fish, so non-bony fish, things like sharks, their genome is a little bit smaller. Reptiles, still smaller. Mammals, super small, right? Amphibians, rather large, and that's because they're pretty complex. They can deal with both terrestrial and aquatic environments. So they have a big genome to help them accommodate that. And then we have flowering plants, which, huge. And again, that's because in terms of biochemistry, because remember, life is nothing but applied chemistry, and plants are the masters of biochemistry. They can do just about everything, right? So it makes sense that they have a pretty large genome. Skill number 1, use of databases to identify the locus of a human gene and its polypeptide product. So let's break this down for a minute. Remember, locus is location of a human gene on a chromosome, and its polypeptide product, right? So this is talking about how we go from DNA to RNA to a protein, because remember, proteins are polypeptides, right? DNA to RNA, this is going to be transcription, and this is going to be translation. That's why we learned about all that stuff first. So what we're doing is we're looking at a database to figure out the address of a specific human gene and figure out what protein it produces. So after the Human Genome Project and the technical ability to sequence genomes, the information that we've gathered has become quite large, and there's plenty of free databases online that you can use to help figure out a particular location of a gene and what it does. So here's one right here. And if I click this link, it'll bring me to a web page, and what I can do is I can search a gene map, right? The gene map is all the information about where genes are located. You can search by genomic region. These are different locations, loci. You can search by chromosome number. So if you know it happens to be on the number 1 chromosome, you can search by chromosome number. Or if you know if it's on the P arm or Q arm of a specific chromosome. And I don't think we talked about this yet. We'll talk about it more when we talk about meiosis. But chromosomes, right, they have a top part, they have a centromere, and they have a bottom part. The top part is the P arm, the bottom arm is the Q arm, and this is the centromere. Now, when you think of chromosomes, you probably think of X's like this, but that's a duplicated chromosome. In reality, chromosomes are just one long part. So this is 2 chromosomes that are tied together. So let's see if I click that. Where does it open up? over here. So let me bring this. I'm not going to donate to OMIM. Okay, so here we go. Here's the gene. This is the database. I just clicked on the link. And here are the chromosomes, right? Pairs 1 through 22, and then your sex chromosomes, X and Y. And let's see. Search by genomic region, genomic location, chromosome, or other searches that will return, for example, so I can do disorder or kinase. Okay, so let's say I want to do insulin. I want to do insulin production. Comes up. All right, so here we go. The location. So this is the P arm of chromosome number 1, right? And the address number is 31, so it's position 31 on chromosome number 1 on the P arm. The gene's name is AIR, and that stands for acute insulin response. So that means if there's a lot of insulin around, I think, acute insulin response. So this is going to be something that helps regulate genes that produce insulin. So insulin is produced kind of it's not produced all the time. It's only produced when you need it. So that's part of the regulatory response there. Here you go. Diabetes mellitus, insulin dependent. So genes that are, if these genes are affected, they can cause diabetes. That would be chromosome number 2, the Q arm of chromosome number 2, and position 24.3. Let's see, if I want to do, let's say, I know, let's do sickle cell, because we talk about sickle cell all the time. Sickle cell anemia. Oh, why did that give me this instead of a table? I'm going to go back. Okay, because I want the gene map, that's why. Sickle cell anemia. All right, here's the table. So these are all the different genes that deal with sickle cell anemia. We have to look at the name. T-cell acute lymphatic leukemia. Ooh, that's a nasty one. Cell cycle division associated proteins, so proteins that associate with controlling cell division. Yeah, pretty cool. So it's basically a map. You type in the gene that you want and it gives you the address. Now when we say address, remember, so if the 1p36 would be chromosome number 1, I can't draw this right now. Chromosome number 1, the P arm at the top, position 36. As long as you understand that you can do this, that's pretty much the point. Okay. So at this point, you should be able to do the following. You should be able to compare the storage of DNA in eukaryotes and prokaryotes. You should be able to define the following: plasmid, homologous chromosome, and histone. And you should be able to compare haploid and diploid cells. You should be able to compare the chromosome numbers in humans with other species. You should be able to outline John Cairns' experiment that led to determining the genome size of E. coli. Now we're going to talk about karyograms. Understanding number 9. A karyogram shows the chromosomes of an organism in homologous pairs of decreasing length. A karyogram is a photo that shows the chromosomes in an organism's cells organized by decreasing length of homologous pairs. A karyotype is the number and type of chromosomes. So here we have chromosome number 1, or pair number 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18. Looks like I cut off the bottom row. You'll notice number 1 is the biggest, right? And further down here is the smallest. So they're organized by length and size. Application number 4. Use of karyograms to deduce sex and diagnose Down syndrome and other diseases in humans. Understanding number 10. Sex. I'm losing my voice all of a sudden for no reason. Excuse me. Sex is determined by sex chromosomes. Autosomes are chromosomes that do not determine sex. And by sex, we mean gender, not gender identity. We're talking about chromosomal assignment. Now, in reality, we know that gender isn't binary. There's a lot of science to support that. But for our purposes, we're going to stick with the XXXY definition of gender for this particular conversation. So within a human karyotype, there's 2 types of chromosomes. There's your sex chromosomes, which determine gender, and that's pair 23, X chromosome and Y chromosome. And then you have autosomal chromosomes that do not determine sex, and they basically determine everything else, right? So that's pairs 1 through 22. We call those autosomes, auto meaning self or body. Just like somatic cells, body cells, autosomal. So the gender of an organism can be determined by looking at the type of chromosomes present for pair 23. Here we have XX or XY. You probably remember freshman year when we did the karyotype posters and you guys got to look up all the weird random problems associated with chromosomes like deletions and inversions and trisomies. That's exactly what we're talking about. We're just going to go through it kind of quickly as review. So, for example, let's look at Down syndrome. So here you have the karyogram of a person with Down syndrome and see if you can figure out what's wrong. Well, I can see it, and I know it. There's a problem here. We have 3 copies of chromosome 21. So if I were going to write the notation for that, the proper notation would be, the first number is the total number of chromosomes, right? So when you're writing a notation, so we have a total of 47 chromosomes, 23 pairs, 23 times 2 equals 46 chromosomes, right? Plus we have an extra one, so that's 47 chromosomes. The second number tells you the second part of the notation tells you the sex chromosomes. So we have an XY. And then the third number tells us what's extra or missing, the location of the extra or missing chromosome. And that would be 21 because it's chromosome 21 where we have the extra. And I would write plus 21, meaning there's an extra 21. So all the information that's contained in this karyogram can be written using 3 numbers. Total number of chromosomes, sex chromosomes, and the location of where the extra or missing chromosome is and whether it's extra or missing. If it was missing, we would write a minus. So let's do another one. Oh, we're going to talk about Down syndrome first. So Down syndrome is inherited, and it's a genetic disease that results from having an extra copy of chromosome 21. This is also called a trisomy. Tri meaning 3. Trisomy. It's a result of non-disjunction during meiosis, and we'll talk about that more when we talk about meiosis. Symptoms include flattened facial features, upward slanting eyes, slowed cognitive abilities, and poor muscle tone. Just a quick understanding of non-disjunction. Here we have a normal cell. We go through meiosis 1. When we go through meiosis 2, we're supposed to split so that each cells are haploid, right? But this is normal. Each cell would get one copy, right? This is not normal. This is non-disjunction, meaning this cell got both and this cell got none. Now, eventually, these are gametes, right? Eggs and sperm. Now, this individual is going to end up with a missing chromosome, right? This individual is going to end up with an extra because you're only supposed to have 2 copies of each. These guys are going to be normal, right? Because they'll end up with 2 copies. We'll talk more about that when we talk about meiosis, but just as a quick overview, when non-disjunction, so the word non-disjunction means to not come apart. Because these chromosomes don't split apart, you get one cell that ends up with both. So junction means to come together. Disjunction means not come together, right? Non-disjunction means don't come apart. Not come together. Okay, Turner syndrome. See if you can figure out what's wrong with this karyotype. Well, 1 through 22 look okay, but there's something going on down here, right? There's only one X. There's not another one. We're missing either an X or a Y. So if we're going to write the notation for this, we're going to count the number of chromosomes. So 2 pairs of 22 is going to be 44, right? Plus 1 is 45. So 45, right? We have 1 sex chromosome, X, and we're going to write minus 23 because we're missing a chromosome at location 23. You can also write XO instead of just X. Either way, that works fine. Oftentimes people ask me, like, how do people with Turner syndrome present? And they present typically, I don't remember actually. You can look it up if you want. Yeah. Next is Patau syndrome. Let's see if you can take a look at what's wrong with this karyotype. Oh, I found it. Looks like there's an issue at location 13, chromosome 13, right? So our total notation would be, let's see, how many chromosomes do we have? 23 pairs is 46, plus an extra one is going to be 47. Next, let's look at our sex chromosomes, X, Y, and then where's the location where the issue is? It's location 13, and I'm going to write a plus because there's an extra one. Okay, let's see if that's right. Yep. Surprise! All right, let's do Klinefelters. Well, 1 through 22 look okay, but something weird's going on down here. So, let's see. 46 chromosomes plus an extra one, so that's 47 chromosomes. We're going to put XXY for our sex chromosomes, and we're going to do plus 23 because we have an extra sex chromosome. I'm right. There's actually a bunch of actors that have Klinefelters. It's not, I mean, it's uncommon, but like you might not necessarily notice by looking at someone. I think Jamie Lee Curtis has Klinefelters, as a matter of fact. Okay, so at this point, you should be able to define karyogram and karyotype And you should be able to describe the use of a karyogram and determining sex and identifying diseases like Down syndrome. I encourage you to go up and look up some more random chromosomal abnormalities, and the symptoms are actually pretty cool. I mean, not cool for the person that has them, but like interesting to learn about. I actually really enjoyed the poster lab that we did freshman year, where you go around and look at all the different chromosomal abnormalities. It's really interesting. Well, that's it for today, folks. Enjoy the rest of your long weekend since we have Monday off. And yeah, looking forward to seeing you guys soon. Talk to you later.